Okay, now we go to your amoebas. We have your Entamaoebia histolytica. Okay, so just remember this. Okay, why does it have a horn like the devil? So it has a horn because they are pathogenic. The other counterpart in the next few slides is the non-pathogenic counterpart. Okay, so Entamaoebia histolytica infects um, 500 million populations over the world, all over the world, okay? And 60 to 70% of the population in Pakistan is infected, and only 1 to 2% in the U.S. Why is this so? In the U.S., you can only see them in refugee camps and military barracks. And they said, I don't know if it is true, but the water source in Pakistan is not very clean. That's why most of the time it is infected with Entamaoebia histolytica. And Entamaoebia histolytica causes amoebic dysentery and liver abscess. Okay? It has two forms, the trophozoite and the cyst. Okay? The cyst is immovable and has a disease-causing form. The cyst survives for a longer period of time because it is double-layered. And the trophozoite, they are motile and have a small shelf life, so they cannot survive outside. They are transmitted by the fecal-oral route. Okay? So you have to remember these 5F. They are the main source of transmission. This includes your finger, fomites, fly, feces, and food. 70 to 80% are transmitted by the 5F or the fecal-oral route. And the remaining 10 to 20% are transmitted by blood. Okay, for all of the parasites that we will discuss, you should at least know the life cycle, but you don't have to memorize. Okay, so for ahistolytical life cycle, the organism enters the host in the form of a cyst, here, mature cyst, which is transmitted through fecal-oral route from contaminated water and food. Okay, the anal-oral route transmission can also occur in male homosexual. There are no animal reservoir. And once the mature cyst is ingested, it differentiates into trophocytes in the ileum. Okay? But colonization occurs in the cecum and the colon. After the trophocyte enters into the colonic epithelium, it secretes an enzyme. As a result, localized necrosis occurs. Lesions, when reach the mucosal layer, causes flash-shaped ulcer, which we will show in the next slide. And when this happens, you will have invasion of the portal circulation. So this is the flask-shaped ulceration in the colon that we have been talking about. So this is your large intestine, and you can see in the diagram, it is shaped like a flask, okay? So for intestinal amoebiasis, you will have your flask-shaped ulcers, abdominal pain, diarrhea, vomiting, and amoebic dysentery, meaning there is blood and mucus in the stool. You can also have extra intestinal amoebiasis, wherein your hepatic amoebiasis and cerebral amoebiasis belong. You can also have mild symptoms, okay, like cramping, nausea, diarrhea. Okay, for your severe symptoms, so the symptoms range from mild to severe. Okay, for your severe symptoms, you will have your profuse diarrhea, liver abscess, severe ulceration, and megacolon amoeboma. Okay? The treatment of choice is your metronidazole or your Flagyl, your tinidazole. Okay, there should be no need to drain hepatic abscess. Okay, you can also use idoquinol or paromomycin for treatment of asymptomatic cyst. For the prevention, of course, you have to avoid fecal contamination of food and water, use good personal hygiene, such as hand washing. Human feces should not be used as fertilizer in crops, and use cooked vegetables in endemic areas. Now, if your Entamaoebia histolytica is represented by a diagram with horn, that means it is pathogenic. Your E. coli is represented by a halo and wings, like for an angel, because they are non-pathogenic. Okay? So what is the difference between E. coli, Entamaoebia coli, and Entamaoebia histolytica is the number of nucleus. When you see less than 4 or 1 to 4 nuclei in the microscope, that is E. histolytica. And if you see more than 4, that will be your Entamoeba coli. Entamoeba coli is a non-pathogenic species of Entamoeba that frequently exists as a commensal parasite in the human gastrointestinal tract. You should not confuse Entamoeba coli with the bacteria Escherichia coli. Okay, so they are two different organisms. This amoeba does not move much by the use of its pseudopod and creates a surplace or non-progressive movement inside the large intestine. Okay? Usually the amoeba is immobile and it keeps its round shape. Okay? So again, you should not mistake E. coli with Escherichia coli with Entamoeba coli. Okay? Because unlike the bacterium, the amoeba is mostly harmless and it does not cause many intestinal problems, just like your E. coli bacterium. Now we go to your flagellates. These are your protozoans that move using flagella, and it's very easy to remember because there are only 3 of them. Okay, you have your Giardiasis, your Trichomonas vaginalis, which is very important, and your Leishmania. Okay, so you have to remember when the exam asks you what type of organism is Trichomonas, it is a flagellate protozoan. So let us discuss first your Giardiasis, and previously known as your Giardia duodenalis or your Giardia intestinalis. It is a flagellated parasitic microorganism that colonizes and reproduces in your small intestine. Okay, this is the organism that you can get from waterborne. You remember in one of our re-recorded lecture, the waterborne diseases, Giardia lamblia belongs to that category. Okay, so Giardiasis does not spread by the bloodstream, nor does it spread to the other parts of the gastrointestinal tract. Why? Because it remains confined to the lumen of the small intestine. Okay, your Giardia lamblia has an outer membrane that makes it possible to retain light even outside the host body. Okay, you can see here in the diagram, it looks like an eye, but these are suckers. Okay, these suckers causes them to adhere to the small intestine for a longer period of time. So this is the life cycle of your Giardia lamblia. Okay, so the human ingests the cyst via fecal-oral route, and then it goes to your small intestine, wherein it multiplies using binary fission. Okay, and then it goes back to the colon to produce a cyst. Okay, so ingestion of 15 to 25 cysts causes the disease. Meaning, if you ingest less than 15, it is not pathogenic. You will not have Giardiasis. Okay, so the eyes that you can see, these are your ventral sucking discs that adheres to the intestinal villi, and the incubation period is between 1 to 4 weeks, with an average of 10 days. 50% of the infected are asymptomatic. Some have mild symptoms to severe malabsorption syndrome. So why do they have malabsorption syndromes? Because the intestine are filled with the bacteria or the protozoans, so you cannot absorb any nutrients that you eat, okay? And then you will have your foul-smelling diarrhea, although they are non-invasive, no blood and pus in the stool, and those who are infected spontaneously recover, okay? If the patient has intestinal diverticula, they will have the more severe form of Giardiasis. Okay, so how do you diagnose it? By fecal antigen test, or when you see cysts and thrombocytes in stools. Your treatment is metronidazole, and prevention, of course, clean waters. Now we go to your Trichomonas vaginalis, another favorite exam question. It is a flagellate protozoan with no observed cyst stage. It is found in urethras and vaginas in women and urethras and prostates in men. Transmission is usually sexual or passage in the birth canal. There are no local invasion and patients are largely asymptomatic. If your Giardia lamblia looks like a smiley face with 2 eyes, this one is one-eyed organism. Okay? So this is how it looks like under the microscope. So this is your T. vaginalis life cycle. Okay. So it multiplies by binary fission. It has no cis stage. So it's always an adult spermatozoid stage. Okay. And then it infects the male and the female through sexual intercourse. And then they multiply by binary fission. And then it resides in the male and female genital tract. Okay. So the clinical syndrome is a watery discharge or vaginitis, intense vulvovaginal itching, and a strawberry cervix. Okay, favorite exam question, strawberry cervix. Okay, it causes prostatitis in male in rare cases. Okay, how do you diagnose it? Usually, you visualize thrombozoids on gemcia or papanicular stain. You will also see a perinuclear halo. You can diagnose it also using wet prep, PCR, and culture. Again, treatment is metronidazole and imidazole. Now we go to your third flagellate, the third and the last flagellate, which is your Leishmania. It is an obligate intracellular parasite that is transmitted human to human or animal to human by a female sandfly. Do you remember this? Transmitted by a vector, which is your female sandfly. In the table below, you have your two types, your Donovani and Brasiliensis. Okay? Your Donovani is the one that causes your visceral, mucocutaneous, and dermal disease, okay, which is common in Asia, Africa, and the Middle East. And the Brasiliensis is common in Central and South America. This is your Leishmania life cycle. Okay, so the vectors are your dogs, cats, and rats. The infected form is the promastigote, and the diagnostic form is your amastigote. Okay, so the Leishmania is transmitted by a female sandfly, and it resides in rats, cats, and dogs. And then, when they are infected, they will develop promastigote. This promastigote will be sucked by the female sandfly again, and then they will invade the macrophage of the sandfly, and they will develop into amastigotes, okay? And then these amastigotes will again be transmitted. So the cycle is repeated. So your Leishmania causes three clinical symptoms. First is your visceral leishmaniasis. So the cycle invade the macrophage of the sandfly, and they will develop into amastigotes, okay? And then these amastigotes will again be transmitted. So the cycle is repeated. So your Leishmania causes three clinical symptoms. First is your visceral leishmaniasis. If you look at the picture on the right, this is common in Bangladesh, India, Nepal, and Sudan. If untreated, it can be fulminant. The incubation is from weeks to years and resembles malaria symptoms. Okay? If you will look at the picture below it, this is your Kala Azar or your black water fever, which is common in India. And then you have your cutaneous leishmaniasis, the picture on your left, which happens between 2 weeks to 2 months from exposure. It is very pruritic and ulcerates. It heals without treatment, but it leaves a disfigurating scar. Okay? And then we have your combination of both. You have your mucocutaneous leishmaniasis, which is caused by Brasiliensis. Okay? And this leaves a disfigurating facial mutilation, just like the child on the middle picture. So how do we diagnose Leishmania? Of course, a clinical diagnosis can be done in endemic areas. And the definitive diagnosis is the finding of amastigotes in clinical specimen or promastigotes in culture. Serology in this situation is not useful. Okay? For the treatment, for your cutaneous, you have your stevogluconate. For visceral, you have your amphotericin B or miltefosine. And for mucocutaneous, you have your amphotericin B.